Hello and welcome to Agri Reflections. In this episode, we'll talk about cytoplasmic male sterility. In cytoplasmic male sterility, in many plant species, occasionally you find strange mutant plants that do not produce pollen due to mutation in their mitochondria. This results in the plant being unable to produce pollen. It can only receive pollen, and since it's inherited via the mitochondria, which is mostly stable, genetically speaking, and is passed on purely maternally, all progeny will be sterile in terms of pollen making. This is used extensively in breeding various types of plants, such as carrot, rice, and corn, to develop F1 hybrids. Here's how a sterile inbred line is created. As you can see by this diagram, you first have the inbred line made through multiple generations of forced selfing to make it purely uniform genetically each generation. What you do is to take that plant and continuously propagate it while crossbreeding that population to the sterile line of plants, and with each generation backcrossing back into the inbred line. If you keep adding pollen of the inbred line in sterile line, the amount of that inbred's nuclear genome replacing the original nuclear genome of the sterile line will increase. If you repeat this long enough, eventually, you will get effectively the entire inbred genome into the sterile line, and you'll always have a good pollen source of the original line to continue to perpetuate the sterile line down the line. To create the F1 hybrid, you find another inbred line that is very compatible with your inbred line and crossbreed it with the sterile version of that inbred line. This prevents self-pollination and makes it easier to have purely F1 hybrid seeds. Now, as you may have guessed, in carrots you do not harvest seeds, so even though the progeny are still sterile, it does not matter since you'll be harvesting the roots. But what if you want the seeds? Like in corn? Well, in this case, many plants have unique mutations in their genome that allows them to deactivate the cytoplasmic male sterility in the mitochondria. These are called restorer genes, and these genes are dominant, so all that is required is for an inbred line to be bred that has homozygous dominant for the restorer gene. When you crossbreed the sterile inbred, you wind up with a plant of seeds, because it will be heterozygous dominant due to the masculine effects of dominant genes. This is how F1 hybrid corn is sometimes developed. Other methods include just detasseling the corn for hybridization purposes. In rice, they use a different method. In rice, the sterility genes only activate in either high temperature situations or under artificially long photoperiod conditions. So in this case, just growing the rice in normal temperatures or environments will deactivate the male sterility in this situation. So the sterile hybrids will be kept in artificial environments to be propagated further, and the F1 hybrid would be grown under more normal conditions. Well, that about covers everything. Thank you for watching.